Good morning, good afternoon, everyone, and thanks for joining us today for this new webinar on proteomics hosted by Affinisep. My name is Florinales. I am the product manager for bioanalysis at Affinisep, and today we have the pleasure to welcome Dr. Aaron Storey from Arkana Laboratories, who will give us an insightful talk on the implementation of the membranous nephropathy typing assay on a triple quadruple mass spectrometer for use in clinical practice. So Dr. Aaron Storey joined Arkana Laboratories in 2023 to direct the mass spectrometry laboratory and is interested in developing and applying mass spectrometry techniques in clinical settings to improve the practice of medicine and address clinical needs. Dr. Storey received his Bachelor of Sciences in Behavioral Neurosciences in 2010 from the University of Evansville and his Ph.D. in Biochemistry and Molecular Biology in 2017 from the University of Arkansas for Medical Sciences. His academic career has provided him with a broad knowledge base spanning molecular biology, neuroscience, genetics, proteomics, statistics, and data science. His skill set combines the application of cutting-edge proteomics techniques to generate large data sets containing high-quality information and the use of programming languages and data analysis techniques to turn this information into actionable insights. Currently, Dr. Storey is working on translating a mass spectrometry-based assay for typing membranous nephropathy into clinical practice. And before we have the pleasure to listen to Dr. Aaron Storey presentation, my colleague Dorian Crevel here will just briefly speak about Affinisep to present our technologies and solutions for proteomic sample preparation. And then it will be Dr. Storey's turn to talk about his research and his work. Thank you all.
Okay, thank you, Florin, for the speaking. So, as you said, I will present you Affinisep, and I will answer about simple questions like who we are, what we are doing, what kind of expertise we have, and what kind of solution, innovative solution we can propose to you. So I will start this presentation about a simple question, which is who we are. So we are basically a technical French company with R&D laboratories and production facilities based in Rouen, so near to Paris. And the most important here is that we are a team of scientists with chemical and polymers specialty, totally devoted to resolve issues from analytical labs and provide innovative solutions. So we have a specific expertise in manufacturing of all our polymeric resin from scratch on our unique technological platform. And here we are able to obtain some specific resin with physical and chemical properties totally adapted to units. So here what we can say is that we innovate for you and also with you. So all sample preparation could be used for different applications, such as, for example, the food safety. Here, with the food safety, we are able, with our Affinisep SPE products, to realize the selection of different food molecules, like glyphosate, for example, patchoulin, and PAH also. And we just developed different MIP, almost 20 different ones, for the selective extraction that we want from our food molecules. Another application will be for the water analysis. So for environmental application, here with our attractive SPE disk and passive sampler, we are able to realize the analysis of contaminants in water, such as pesticides, drug residues, endocrine disruptor, for example, and also PFAS. But here, the most interesting application that we can talk about for this presentation will be a biological application with bioanalysis and proteomics applications. Here, I will talk really quickly about BioSPE ready to use, because I will go in more details later in these presentations. But for our BioSPE products, we can just apply it for various applications in proteomics. But as I said, I will go deeply for this application later. For now, I will talk about our SPE membranes and specifically what kind of advantages you have to use our SPE membranes. Here, our membranes is a thin membranes made from small sorbent bits tightly incorporated in the whole membranes, and we have a high capacity for the extraction and purification of molecules of interest. Some advantages here to use those membranes. First one will be that there is no need to use fluids or filter to maintain your membranes, and so you will have some reduced dead volumes, and it's simply to use. In terms of efficiency, you will have an excellent repeatability and a high sample recovery, in addition to the fact that you can use those membranes with small elution volumes. This allows you a shorter evaporation time and also an efficient concentration of your analytes. In general, those membranes could be used with a wide range of sorbents, and it's a first point in terms of custom solution that we want to propose to you. The second point will be about our formats. We have 4 different formats for the microelutions: the chips, the spin columns, the well plates with the 96 and 384 wells, and also the cartridges. For each format, we just propose you a different size, depending to your working volume, and different capacities of retention. So this format represents a second point in terms of customizable solution that we can propose to you in addition, so with our sorbents. I will be more focused now about the chips and how we can just produce it and how you can use it. So first point, how we produce it. We used a fully automated packing method, which permits no type-to-type variability and a highly repeatable process. And thanks to our Jack automats, you will not waste some time to do your stage chips by yourself, because Jack will do it for you. In terms of use of our chips, here we just conceived and developed chips for optimal use by centrifugation. Advantages with centrifugation will be that it's less tedious and less painful than manual peptide. You will have some more repeatable results, and you will save some time by centrifuging several samples simultaneously, for example. So now how to use it? It's simple. When you just buy some ready-to-use kits, you will have chips that you need to incorporate in a spin adapter, and after in the collection chips. You introduce your liquid on the top of your chips. You let centrifugate it and let your liquid pass through the sorbent, and you just have to collect it to analyze it. If you just want more information about how to use our chips, I invite you to watch our video on our website. If you don't find it, you can just send us an email and we will provide it if you want. So you can use our chips by centrifugations, but there is another technique, which is a positive pressure. In the same way, you just have to introduce your liquid on the top of the chips and apply your positive pressure to make the solution pass through the sorbents. And thanks to this possibility, you can use our chips in a fully automated workflow, which different kind of automates, like GGS Pro from CEM, the Resolvex A300 from TCAM, and also the EvoSep ones. So here I was focused about the chips and how we can use our chips, but I can talk a little also about our other formats, such as the spin columns, for example, for high peptides and protein amounts. Here, all spin are simply to use, and you can, for example, close it for storage some sample or incubate some ones. For our plates, advantages to use our plate is that you can use it partially if you want, and you have multiple methods of use, such as centrifugation, positive pressure, and also vacuum manifolds. For the cartridges, for large sample volume, you can use it by positive pressure and or vacuum manifold. And if you want just to pass through a large sample volume to small one, it's totally possible with this kind of formats. So I talk a lot about formats. I can talk now about our sorbents and the chemistry around our sorbents. Here at Affinisep, we propose you two product range based on our SPE Membranous technology. The first one will be the Attract SPE disc with a wide variety of sorbents, such as silica-based sorbents and polymeric sorbents. So here you can use this kind of product range depending to your chemistry, for example. Another product range that we can present here, it's the BioSPE. I already talked a little bit about this one. It's a ready-to-use kit, specially developed and designed for different applications in bottom-up and top-down, so in general in proteomics here. We can talk about peptide desalting, peptide fractionation, glycopeptide enrichments, removal of magnetic bits after FP3, and also purification of intact proteins. For this presentation, we will be focused about our BioSPE products and specifically about peptide desalting, because around Storey will present it a little bit in his presentations. So for the peptide desalting, we can talk about BioSPE PurePep, and here we just use our BioSPE PurePep tips 200 microliters for large desalting, and we obtain this result. Here you have in yellow box the references and blue box our results. And as you can see here, the result that we obtain compared to the reference are really close in terms of identification. So it means that we have a high capacity to identify our proteins. Also, we have a good recovery of peptides on a wide polarity range. If you just want to purificate different peptides, for example, native peptides, phosphopeptides, glycopeptides, or TMT labeled peptides, it's totally available with this product. And last but not least, we have also an excellent repeatability in our results. Another BioSPE product that we can talk about is BioSPE PurePep Broad. Here, if we compare it with our PurePep BioSPE, you can use the PurePep Broad with for a different polarity range. For example, with highly polar peptides, it's totally suitable with this kind of products. And we have some difference and some advantages also to use PurePep Broad. Depending to the polarity range, you will still have a high number of protein identified and also a an excellent repeatability. If you just want to work in an extreme pH condition, or if you just want to run dry during your SPE procedure, it's totally available with PurePep Broad in the opposition with PurePep. But if you work with TMT labeled peptides or phosphopeptides, you are more suitable to use the BioSPE PurePep. So if you just want more information about our results, I just invite you to watch our application notes on our website. If you just want more information about our other projects, for example, the C18 or BioSPE PurePep Frag, we have different posters if you just want to see it on our website. And if you just want to meet us, I invite you to come to our booth, to EPA for the next week, or if you're in GFSM in Montpellier or Liverpool, it will be a pleasure for us to meet us. And so I will just thank you for your attention. If you just want to contact us, you have our address email, so Florian and mine. And if you will have any question, it will be a pleasure for us to answer at the end of our own presentations. Thank you very much.
Thank you, Dorian, for this quick overview of what we do at Affinisep. So like Dorian said, if you have any question about our products or technologies, you can write them down in the question and chat tabs, and we will answer them after Aaron's presentation. So now I will let Dr. Storey talk about his work and his study on the implementing of a membranous nephropathy typing assay on a triple quadruple mass spectrometer for use in clinical practice. So Aaron, the floor is all yours. Thank you.
Thank you, Florine. Thank you all for attending, and thanks to Affinisep to give me this opportunity to talk about what we're doing here in the mass spec lab at Arkana Laboratories. So we are about to roll out an assay that is set to classify membranous nephropathy using mass spectrometry. If you can see my mouse, we're going to be looking specifically at these electron-dense regions in this EM image of biopsy tissue, and the focus is these immune deposits that are causing the disease. This project is the product of a long collaboration between Arkana Laboratories and the proteomics core at UAMS, the University of Arkansas for Medical Sciences. The proteomics core was rebranded to the IDEA National Resource for Quantitative Proteomics in 2020 after they secured an R41 grant, and the mission of that grant is to provide the highest quality proteomic services at the lowest possible cost, which in our mind, or their mind, it's the biggest barrier to proteomic services is the cost of doing proteomics. So that's the proteomics group, and I learned everything I know about mass spectrometry from this crew. And the other side is Arkana Laboratories, which is the largest nephropathology practice in the world, as far as I know. So this company was founded in 2003 by Pat Walker, and it was founded on the principle that there is no greater length of time between when a patient has a biopsy taken and when they hear back the results. So based on this understanding and based on the commitment to address this need, Pat founded this company to provide same-day biopsy turnaround for renal biopsy tissue. So on any given day, Arkana Laboratories receives about 120 kidney biopsies in the morning by FedEx, and these biopsies are accessioned, processed, stained by, or analyzed by various microscopy techniques, and these results are reported on in that afternoon. So if you send Arkana Laboratories a biopsy today, you will get a call back tomorrow to go over the results, tomorrow afternoon to go over the results. So I started working with Arkana when I joined the proteomics core as a postdoc in around 2018, and to study a disease called membranous nephropathy. So this is a distinct kidney disease that is driven by autoimmunity. Although that wasn't known at the time the disease was given a name. It was first described in the early 50s by David Jones after inventing the Jones stain. And he noticed that in this particular disease, there is a thickening of the glomerular basement membrane. So when you hear membranous nephropathy, it means thick basement membrane, thick membrane. So that was first described in the 50s. And a few years later, it was determined through experimentation that these thick membranes are enriched in immunoglobulins, the immune molecules that attack specific proteins based on their affinity in their variable domain, variable region. So it was known in the 50s that there's a thickening of the basement membrane and that these membranes contain immunoglobulins, but it wasn't understood why that is for several decades. But that changed in 2009 when Larry Beck and his team reported that the primary target of membranous nephropathy in the majority of membranous cases is the protein PLA2R. And they did this using a brilliant experiment that used a combination of serum, sera, and kidney tissue from affected patients. So I'm using hypothesize as a narrative device here, but they hypothesized that since these patients have thick membranes that are full of immunoglobulins, these immunoglobulins could be coming from the blood. These could be immunoglobulins that are attacking proteins in the kidney. So that was, if that hypothesis is correct, if these patients have specific immunoglobulins in the blood that are attacking some protein in the kidney, then this Western blot experiment should reveal what protein is being targeted. So they ran kidney proteins of the affected patients on a gel and used the serum from the affected patients to probe for what protein is being attacked by the autoantibodies. And when they did this, they found a common band across several membranous cases that wasn't found in diabetic nephropathy or other kidney diseases. And doing a gel CMS, or taking a gel CMS approach, they identified the target protein to be PLA2R. They further showed that many of these patients have autoantibodies in their serum that react against PLA2R. So that revolutionized our understanding of membranous as an autoimmune condition. And using the same kind of approach, another antigen was identified a few years later. And since then, there have been around 30 antigens with varying levels of clinical evidence showing that these are target antigens in membranous nephropathy cases. So why does that matter? Why is it important to know the target antigen in membranous? And the reason is that if you know what protein is being targeted by autoantibodies, you can then look for these autoantibodies in the blood by using ELISAs, or mostly ELISAs. And it's been shown that the autoantibody levels in the blood rise before disease flares occur. They correlate with proteinuria that are more later indications of the disease. You can monitor whether or not a treatment is working because the autoantibody levels in the blood should fall before the kidney injury is fully resolved. So it allows for less invasive or non-invasive monitoring of the disease by measuring the autoantibody levels in serum. But it's necessary to know what specific protein is being targeted. But another reason to look for the antigen is that it's been known since the disease was described that it sometimes happens under contexts that flare up the immune system, driven by an immune response. So things like cancer, malignancy, autoimmunity like lupus, or infections, these can trigger the immune system, and if that goes awry, it can lead to immune complexes that clog your kidneys. But what we're seeing now are indications or associations of specific proteins being targeted and specific disease contexts. Some of these, it's more complex and broad, but some of these proteins have more specific associations. For example, contactin-1 membranous nephropathy is associated with a neural condition called chronic inflammatory demyelinating polyneuropathy. And this particular protein is found at nodes of Ranvier, and this can get targeted by the immune system, and then these immune complexes can ultimately clog kidneys. So knowing if a case presents with contactin-1 membranous, that can reveal an underlying condition that may be involved in disease formation or progression. So this second reason is that there are disease associations between the protein being targeted and the root cause of the disease. It's really exemplified in two cases, or I guess my two favorite examples are a mercury exposure outbreak and then NDNF and syphilis. So there have been reports, a couple different reports of NEL1 membranous nephropathy that is linked to mercury exposure from counterfeit skin whitening creams. So in these reports, a lab noticed 3 patients who had NEL1 membranous nephropathy, and it was determined that these 3 patients had used the same skin whitening cream bought from a secondary market. They tested for mercury levels in the cream and found high levels of mercury. And in 1 patient, at least, there were high levels of mercury in the blood and urine. So by identifying that through NEL1 nephropathy that was linked to mercury exposure, they were able to identify the root cause in those patients. And by not using the skin whitening cream anymore, actually, I don't know if it resolved, but it's may have been resolved. So some of these associations, these disease associations, are reversible. And if you can deduce, or the pathologists and nephrologists can deduce the root cause, that can inform treatment. Other case is NDNF nephropathy and syphilis, neurosyphilis. This was described a few years ago, and for reasons or mechanisms that we have yet to understand, syphilis infections can reach the brain, and when that happens, it can lead to a brain protein, NDNF, in order unknown, but NDNF leaking out of the brain, and given that the brain is immunoprivileged, this can be recognized as a foreign agent by the immune system. And what this means ultimately is that if a patient presents and has NDNF nephropathy, that can raise suspicion for an underlying syphilis infection, and that can be followed up with an RPR test or a test for syphilis. It's not always syphilis when NDNF nephropathy is found, but there is a strong association between NDNF nephropathy and an underlying infection. And again, this is treatable by intravenous antibiotic regimens, which can potentially resolve the kidney symptoms. So being able to monitor by non-invasive procedures and by being able to identify the underlying context that's driving the disease is why we're doing this. But since 2009, there have now been so many antigens described in the literature that it's led to a proposal for a new way of classifying the disease. So the Mayo Clinic got together the world's leading experts in nephropathology, and they came up with a scheme or an approach or guidelines for how membranous should be classified. And they proposed a two-step process of first determining and identifying the target antigen in that case, and then commenting or reporting any disease associations if they are present and known. But that presents a challenge to nephropathology labs, because there are now so many possible antigens that could be driving the membranous nephropathy in a given case that it's impractical to test using single point tests. And also there's a limited amount of biopsy tissue. So that forces the nephropathologist to use these complex decision trees in order to make the most out of the material on hand and hopefully find the target antigen in a given case. And they can use the disease associations in order to make the best guess for what it could be. And it really is amazing to see that working. However, we think that this process can be simplified with a multiplex assay. So what we proposed in 2020 is that since these antigens almost all have been identified by mass spectrometry, perhaps we can develop a diagnostic assay using this technology, using a pull down of the immune complexes using protein AG beads, and using targeted mass spectrometry to look for these specific proteins that could be the target antigen in a case. Oh, and I did not mention at the beginning of this that this work was supported or made possible through an STTR phase 1 in 2021, and then an SBIR phase 2 in 2022, going up to the grant just ended last month. So this wouldn't have been possible without the support of the NIDDK to support this work. But in phase 1, we asked that question: can we do an immune complex pull down and mass spectrometry to identify the target antigen? And for this was a still discovery type, semi-discovery type approach, but we used DIA as a targeted method on immune complex pull downs that were then subjected to bottom-up proteomics to see if we can detect the target antigen. So we took a machine learning approach. We ran 300 samples, about 50 PLA2R positive, 50 thrombospondin, 50 dextrose, and then 50 or 125 negative cases that were negative for these antigens, and then tested if we can build a machine learning algorithm or a number of machine learning algorithms to classify it based on the mass spec data. And what we found is that yes, you can implement machine learning algorithms to do that. The signals are strong enough that yeah, you can teach a machine to do this. Linear model works well too. I mean, the signals are strong enough that you can employ a number of algorithms to do this. So in phase 2, we wanted to turn this into a clinical assay for use in clinical practice. So to do that, we needed to build a lab. So this was the state of the lab as of April 2023. So over here is the the molecular department at Arkana. And over here is the revenue cycle management and materials management department. But this is what the lab looks like now. Over 2 years, we've installed the instrument, put in all of our supplies and equipment needed to process tissue, and we got to work. So after lots of experimentation, lots of optimization, we have landed on this workflow as the simplest approach for getting this information. So we start with either tissue sections or the residual core block from from cases that have been fully worked up. And we perform a lysis procedure to solubilize immune complexes. And then we perform an immune complex pull down using protein A G beads. We also spike in 100 nanograms of an isotopically labeled immunoglobulin so that we can, as an internal control for the pull down. And then after doing the pull down, we digest with trypsin. And these two steps, these two processes are implemented on a Kingfisher Flex 96, which is a solid particle processor that can move magnetic beads through a series of trays. And this can be used or utilized in order to carry out a protein or an immune complex pull down, and then a digest by SP3. So after digest, we add in our internal standard peptides, and we clean up using Affinisep's BioSPE plates. We use the broad pH, high capacity 96 well plates because they work quite well. And yes, the small elution volume we found to be very useful for the assay because we're also committed to getting this information as quickly as we can, and we've gotten to a workflow that works within an 8-hour period. So we can process tissue and get to samples that are queued up on the triple quad overnight. And the low elution volume, we can elute in 80 microliters of 70% B, and that reduces the drying time to a level that works in our workflow. And when we first built the lab, we tried our normal desalting resin, but also tried the Affinisep resins. And we found when doing analytical studies with our synthetic peptides that peptides that contain alkylated cysteine residues are recovered much better on the Affinisep desalting plates compared to other desalting products. So this is spiking the synthetic peptide into the dry down or into the elution material. So just spike into the end of the process. This is spiking in before desalting, and same with here. Desalted on a setback plate for a setback resin versus Affinisep. And we found that we get much better recovery for the majority of our peptides in the panel using these plates. So from there, to build out this assay, we wanted to get every step of the process worked out, optimized, and under control. And to do that, we followed the guidelines by recommended by the Clinical Proteome Tumor Analysis Consortium, CPTAC. They've proposed guidelines that that follow the principles of analytical chemistry, and they are great preparation for developing clinical assays. They propose a series of experiments that go from establishing an analytical measuring range by doing a dilution curve to get linearity and sensitivity, and then verifying this range by a number of experiments across different laboratory conditions and under different matrices. And it ends with an experiment to detect the endogenous analyte in the whole measuring process. So we followed that last summer after we got our synthetic peptides, our isotopically light and heavy peptides, so that we can model these responses according to our analyte quantity and see if it follows a useful model or a linear model. And so by following this experiment, you can derive a response curve that tracks instrument response for a given quantity of analyte. And in doing so, if you have this carried out under known quantities, then if you have an unknown sample within that range, you can ascertain the quantity of that analyte based on instrument response. So we did this and found that our limits of detection are in the femtomole, some subfemtomole range. But yeah, follow these guidelines to obtain these initial figures of merit to see how well this could work in practice. We did find that the more hydrophobic peptides that elute later are less reliable as quantitative surrogates. So many of these peptides that have lower R-squared values have since been removed, not for this first experiment, but I actually wish I would have dropped some of these earlier. We found that they're not worth the cost of maintaining them in the assay. But skipping through a couple sections through this analytical verification process, the last step is testing if we could use these isotopic peptides to detect a protein from a digest. So ideally, we would do this on defined material that we can pull out of solution, but that's impractical. The best that we could do for this experiment is spiking in recombinant proteins into the immunoprecipitate and then asking, can we consistently detect these proteins in our workflow? So this is spiking recombinant protein into our IP plate and then testing if we can digest this 5 different times, 5 different days, triplicate, and what's that signal look like? And we found that this Kingfisher digest workflow reliably yields signal for our target proteins when they are present in the sample, and they are not detected in samples that do not have this protein. And for some of these proteins, it turned into more of a measure of protein freeze-thaw stability than quantitation itself. But we found that this is a consistent and reliable method for detecting proteins from samples in this Kingfisher workflow. So that was our analytical verification, but we then wanted to apply this to unknown samples and see if we can detect the target antigen in these cases. So we've over that course, we've taken a number of data processing workflows. Ultimately, the mass spec signals are strong enough and specific enough that we do see a signal for the target antigen in pull downs of residual core lysates of these patients. So that was using a combination of Skyline and exporting the peak area ratios. And yes, we can see the specificity and selectivity through this, but we'd like to have this in a way that is more reportable in a clinical setting. So for that, we're using TraceFinder, which is designed for mass spectrometry in a number of fields. I believe food safety and wastewater analysis or various industries, but for clinical reporting as well. It's designed for reporting results in a way that complies with the various guidelines for different industries. And in doing this, I will say it's nice to see when we detect a protein that we see that the quantities, the estimated quantity based on our calibration, estimated quantity for these peptides are highly correlated or tend to be very similar in pointing to a protein. Like this gives us some confidence that we have multiple peptides to a protein pointing to the same quantity of that protein. That's encouraging. So over the last couple of months, we've been in the pre-verification stage to see if we can use this to detect antigens in either typed or untyped cases. So I'll preface this by saying that this is still preliminary. It's not peer reviewed yet, so grain of salt. But this is what's been going on in the last couple of months. So we've worked on. We used 2 different cohorts in pre-verification. One was 159 cases that were negative for 4 canonical antigens, PLA2R, thrombospondin, exostosin, and L1. And another cohort we processed and analyzed because, and the data for the second cohort is down here. These had a number of PLA2R positive cases, but the reason we used this cohort is to work with material that has a low number of glomeruli in the sample. So, so samples with limited glomeruli, limited biopsy material. So that was a way of challenging what is our limit of detection with this workflow. And again, preliminary, but soon, yeah, this is not peer reviewed yet. But when we did this experiment, we found by mass spectrometry detection of 20 distinct antigen proteins in the pre-verification cohort. So this is both cohort 1 that had the quad negative cases, as well as cohort 2 that had many PLA2R cases, some of which with very low number of LOM counts. For the cases that were negative for canonical stains, we found a mass spectrometry signal mapping to one of these proteins in about 40% of cases. And we're almost finished with doing confirmation staining on these. I believe almost all have been confirmation or verified by immunohistochemistry or immunofluorescence. There are a few remaining groups to still do confirmation staining, but we are in essence calibrating and finding what quantities of detected peptides for these antigen proteins correspond to positivity by IFIHC. So in a way, we are calibrating this assay. We're now calibrating this assay against IFIHC stains that we would, the information that we would get from those tests if we did that on 28 stains. And it's going to have to show a couple images for these recent detections. This is for CRIM1. It's a relatively new antigen that was described sometime in the last 5 years. We hadn't detected it previously in Arkana because we didn't know which cases to run this on. But based on the mass spectrometry data, these screening results, we found that by confirmation staining that these cases have CRIM1 present in the glomeruli that is presumably causing the disease. So we see glomerular staining of this protein, I think okay, I can't comment on the exact location, but the pathologist said they were positive, showed the images, and they look really nice. So our point being that our mass spectrometry results, we are calibrating these against immunostaining to essentially to get the same information that we would get if we ran 28 immunostains on these cases. And we're also interested in automating the data analysis process. So we can use Skyline to analyze this data, but we want this to be established, performed, and maintained in TraceFinder in a clinically compliant and workflow. So we are working on configuring TraceFinder to output results that are concordant with the IFIHC results. And we've done some analyses to figure out under what experimental conditions do we get confident results, or not just confident, but concordant with IFIHC. What we found is that if we can recover enough Silamab signal, which means that the pull down worked and the desalting worked, Silamab means that the pull down worked. But also, so the x-axis here is the amount of Silamab peptides, the heavy labeled IgG, but the y-axis is the endogenous IgG, the immunoglobulins that are causing the disease. And we found that if we can recover enough IgG from a sample, more than 100 nanograms, we get an answer that is largely concordant with immunostaining, IFIHC. In this case, the EXT2 cases here, that was due to a quantity cutoff that was too high. So we're working on the parameters that yield an answer that is concordant with IFIHC for these antigen proteins. So that's where we are in the lab. And we found that we can break this down into domains, and with this 80% accuracy, we're almost there. There are things that we can where we're still improving the configuration, and we are sorting through. Currently we're at 80%, when we have captured enough IgG and captured enough of our internal standard. So that's where we are in the lab. We're eager to, we've been very encouraged by the results, and we're eager to get this running in clinical practice. And we are figuring out the best way to get this into practice as quickly but as accurately as possible, in a way that's efficient. So again, thanks for your attention. Thanks to the NIDDK for their support. This wouldn't have been possible without their support. And thanks. Happy to take questions.
Thanks a lot, Aaron, for this presentation. It was very interesting. So if you have, I'm talking to people attending this webinar, if you have any question that comes to your mind after, or if you think about something, you can also write the question to Dorian or myself, and we will then transfer the question to Aaron so he can answer you if you think about something or later. So don't hesitate, we can put you in contact with Aaron if necessary in the future. Okay, well, I'd like to thank you once again, Aaron, for this very interesting talk and for taking the time to present your work during this webinar. It was a real pleasure. And thanks everyone for attending. And so like Dorian said, we will be at UPA next week in Saint-Malo. So if you are attending this event, you can also come visit us and talk about our different products there. And if not, maybe we will see people and at other events or other webinars. So keep informed, and you can subscribe to our newsletter for more information about our upcoming events. Thanks a lot, Aaron, again, and I wish you all a good day or a good evening, and see you soon. Bye.
Thanks a lot. Bye, Aaron.
Thanks again.